Okay,
Did you start it?
yeah.
Okay.
You ready?
No, yeah, go ahead.
Okay, so this next class 10 talks about 3 papers. One is Zhang's paper on regulation of CCPA, and he's done this using RNA-seq. This paper's in PLOS 1. Bernie's from Florida. The second paper, Patengi. Let's take a look. So Patengi, where is he? Looks like he's from Germany. Institute of Medical Microbiology. That's where the Pyogenes paper comes from. Then the third paper, Transcriptomic and Genomic Evidence for Agalacticae, is written by a guy by the name of Richards, Vincent Richards. So this paper's in BMC Genomics, and they're looking at Streptococcus agalacticae. Agalactiae? Yeah. This is done in the lab of Michael Stanhope. Stanhope's pretty good at doing RNA-seq work. He collaborates with Robert Burney. So the first paper, like I said, was the Zhang paper on catabolate repression. Now, just as a background, what is RNA-seq? This came from Wikipedia. So what you do is you take your total RNA, isolate it, fragment it, and what you could do is, say, add poly A tail to all of your RNA. And if you do that, you can use magnetic beads, and magnetic beads with the poly T tails. So you get left with magnetic beads with lots of RNA on the outside of it. Then what you do is you isolate those magnetic beads, and then you fragment them and put them onto an Illumina or Roche or solid sequencing format. So you get all of the RNA sequencing data.
Okay.
So that's how they did this experiment, the sequencing part. So it's essentially very similar to a microarray. The only difference being that in a microarray you have your specific gene regions, or you're actual hybridizing 2 regions, where this is just the RNA.
Yeah, you cannot get in microarrays, you cannot get non-coding.
You can.
You can?
You have to make the region, though. You have to actually design the microarray because the microarray that we would normally do is design just for the genes. You'd have to design it for the genes and the intergenic regions too. All right, so one of the things that comes out of this paper is Bernie's predicted a number of small RNAs. He's got in his list, he's got over almost close to 300. However, if you look closely at the list, a lot of them are in RNA regions. So they actually figured out the small RNAs come from ribosomal RNA.
Oh, so, you know what? Because they
didn't get rid of the small
RNA stuff. There is a new technique. Remember the paper I presented? They use the tags. So actually, it's an exonuclease terminator. So you get rid of all the ribosomal RNA,
which is great.
Yeah. So they didn't have that.
No contamination. So this data is good, but it's also got a lot of ribosomal data in it too. I'm going to show just the tables first. So this is the small RNA table. Here's a table showing that they've done differential expression of UA159 and their CCPA mutant.
Okay, CCPA
is a catabolite repressor protein. It is involved with the GTFs of S mutants. So it's really required for knowing when nutrition or glucose is available to the cell. And this is why they're doing it in
so carbohydrate,
glucose.
In the presence of carbohydrate.
Right. And this paper carefully looks at two different kinds of carbohydrates. They're looking at glucose as well as galactose. So this table represents their genes found in glucose. So if you take a look, this is table 2. You can see, oh, this one, the kinds of genes we're looking at, dehydrogenase genes, and there should be GTF genes here too. PTS genes are there.
Yeah. Glucosyltransferase, yeah.
So, and what they're also showing is, okay, so some of these are in bold. If they're in bold, they've been shown before in a microarray to be upregulated, or it matches what they see in the microarray. If it's not bold, this is new information. So a lot of this is actually brand new information that they're getting using this technique. Okay, next table, table 3. So they're looking at the difference between glucose and galactose. And this one you can see a lactose repressor, galactose 6-phosphate, PTS systems. There's lots of sugar genes here on table 3. And again, the ones in bold were known before, and there's some that they're just figuring out now because they haven't done this kind of work before. I didn't show the whole table, I just showed part of it. Next, table 4. Now, if they're looking at the mutant, they're looking at the expression of that mutant. So they've done work with microarray of this mutant before, and they're just confirming that a lot of the hits that they've had before are exactly the same that they're getting now. And these genes, for the most part, are very similar to the ones on the page before because they're doing a very similar experiment.
Okay. Comparison between glucose and galactose.
And in this 5th table, they're looking at differentially expressed genes between mutant and wild type in galactose. So the specific sugar galactose, they're not just looking at glucose now, they're looking at just galactose. Because that sugar has a different profile than the other sugar. Okay. Now, in the figures, the first figure, what they're showing is how to cluster their experiment. Now, TW is the deletion mutant, and what they've done is they've done a number of runs, glucose with TW. So they've done it in triplicate, glucose with UA159, galactose with TW, and galactose with the UA159. What they're showing is when you compare the results, with glucose TW1, those 3 experiments pair up very nicely. Glucose TW1, glucose TW1. It's almost the same experiment.
No, it's the same, Andrew.
Pardon?
It's the same.
Yeah. And this is what you would expect. you want a nice straight line down the middle, and you want glucose to match glucose. You want UA159 to match UA159. So they're showing that they get a good confidence in their data.
Oh, okay.
Okay? Their data clusters nicely together.
So this is enough to confirm?
Well, it's a lot of data in just one small image, just to show that it's good data.
Heat map, huh?
Yeah. Next, what they're doing is they're showing you the frequency of the transcripts, so they can come up with different values for their transcript. And the values, as you can see at the bottom, the expression level 36, 180, 320. So 36 shows that there's low level of expression. They're going up to 26,000 high level of expression. And the number of transcripts at 36, you get close to 200 maybe, and you get a range of everything in the middle. And then 2600, you get about 100 of those. Now, these are all predicted transcripts. They're not novel ones. These are ones they expect to find. Next, this is an interesting scatter plot. What they're doing on the left in A is they have two genes that are in an operon, and they look at the sequence. And they say that the transcription of the two genes in an operon are transcribed together. So that forms a really nice straight line. Now, if you have two genes next to each other that are predicted not to form an operon, they just form a big blob. So this is good data. This shows that that's the expected result. Then what they do is they find out all of the classes of genes that have a large change, and there's a very long list of these genes.
Oh, isn't it?
Same thing with their, so this is done on glucose, and then they do the same thing with glucose and galactose.
Functional classes of differentially expressed genes.
Mm-hmm. And on the bottom, they're also reporting the log2 fold change so that you can see, okay, so energy, metabolism, sugars, which ones have a large log fold change. So they found these ones. And in order to see which ones those are, you'd have to go to the supplementary data, because there's so much data here.
Yeah, they just try to put this all together in a paper.
Yeah. And then the next one, it's the difference between glucose and TV galactose.
I don't have this one, right? Is this one?
This is the last picture. Yeah.
Mm-hmm.
Okay. And that's our paper.
Mm-hmm.
So it's not really informative about the gene regulation. You'd have to go through each and every gene to figure out.
And they need
what's
to
going
confirm anyway. So this is just data.
Right.
That,
This is the
the platform. So you need to go if you want to work on something, which is a gene.
The next paper is, I think, better, because what they're doing is they're actually doing confirmation of some of these small RNAs. Mm-hmm. Okay, so the next paper is on pyogenes, M49, and they're looking at a tiling microarray. So here's a picture of a tiling microarray. What you have is you have the genomic DNA, and you could do this one of two ways. You could have the hybridizing regions overlapping, or in a row. And this could be genomic DNA that's intergenic or genic DNA. That's what the idea for a tiling microarray is.
Mm-hmm.
And so in this paper, they have a big table showing you a number of sRNAs. Now, a number of sRNAs have already been identified, like FASX, and there are really important small RNAs that are being investigated, but here's a fairly well organized list of new ones that they've come up with. Ones that have a name would be previously identified. Oh, I don't see one that's here. Oh, here, previously published. It's this column here. So a number of them have been published before. 1, 2, 3, 4, 5, 6, yeah, about 10 almost. Oh, and there's the FASTX right at the top. So what they're doing is they're confirming that these intergenic regions are expressed. So in the next table, they're showing differential regulation depending on media. Also, it depends on the growth phase. So you can show how some media control the expression and how, this is table 2 that I'm showing. Probably closer to the end. Yeah, I went over the tables first, then I thought I'd show the pictures next. So in column 1, they have THY versus CDM. So they're showing if you have starvation media, which ones are upregulated and which ones are downregulated. That's in the exponential phase. The next column, stationary phase. So rich versus starvation media in stationary phase. And they found a number that are downregulated.
So some of them are in all phases.
Yes.
All growth phases.
So this would mean that they had to do this tiling experiment in THY and CDM as well as BHI as well, and also look at stationary versus exponential phase. So it's a lot of different conditions they've had to look at.
Well, anyway, there is not a big change between, so they are constant. They are expressed all the time.
Some are, and some are different. Some are very different. Some you only see once. Like the very top one you see there at negative 2.72, it's only downregulated at stationary phase when you're comparing THY and CDM.
Okay. CRISPRs.
Yeah, they looked at everything in this paper. So they noticed that they're there. The next picture is kind of interesting. I like this sort of map of the Streptococcus genome. And the reason I like it is because every time you look at a Streptococcus genome, you see this picture, this half and half. It's always divided up into 2 parts. Doesn't matter which Streptococcus you're looking at. And what they're doing is just mapping out the GC content as well as the forward and the reverse genes. So track 1, forward reverse, track 2, GC, 3, 4, 5. So track 5 are the small RNAs, I think. Tracks 4 and 5, sRNA genes forward and reverse. I like these maps.
You like them?
Yeah. They give a lot of information just right in a very, very condensed way.
How do you get these ones? This representation?
You would have to
upload your raw data.
Put your data into McVector. And you'd have to kind of identify all of the sRNAs and make that as another circle. In the next one, what they're doing is they're looking at a number of sRNAs and trying to figure out where they start.
Skipping something?
Right here.
Oh, okay, sorry.
So you can see FastX, they're just identifying the start site,
Oh,
as well as the coding region.
okay. This is what you did, right?
Yeah. The next picture is not quite so interesting. They're comparing their result with bioinformatic results. So in this study, see they've got 38, 5, 1, so that's 43, 44, 55 regions identified in the study. And if you do it bioinformatically using these two other methods, they give you different results. So depending on how you do it, it'll give you different results. That's what they say. That's what it's like. And there is another tiling array here. So in this study, they have 43 identified that are unique and different than the one that was done in M1T1 by a different group. So another group has done a tiling microarray as well.
And they found 12 in common.
12 are the same.
Mm-hmm.
Interesting. Now, if you take a look at a couple of candidate sRNAs, you can figure out if they have a motif or not. And you're familiar with the motifs. And this is something that you can do by aligning 2 or 3. Well, in this case, this one has 2, 2, 2. They'll have 2. A minimum of 2 for the motif. Oh, and this one, here's a T box. There's L10 liter. CRISPR. I thought you'd like that. They figured out the CRISPR motif for S biogenes. And then what they do is
CRISPR motif for what?
So there must be a CRISPR that they're looking at in this region.
Yeah, it's probably the repeat.
Could be. You'd think there'd be 5 or 6 or 7 or 10, not just 2. Well, based on how they do the tiling microarray, they might not have found all of them. They should know that
But
they're there.
You can just go to a CRISPR finder.
Yeah.
Yeah, and you just upload your sequence.
Next slide, what they're doing is they're taking a look at those small RNAs and coming up with expression values. And this is how they're doing it. They're looking at the different media, CDM versus THY, also OD 1.2 versus 0.4. As you can see, when you're looking at different media, CRISPR-1 pops up, showing that in starvation media, it's upregulated. FastX is around 1, and these other ones are slightly repressed. As opposed, when looking at exponential phase versus stationary phase,
in stationary phase,
CRISPR-1 is more abundant in stationary phase than exponential phase.
Yeah.
Same with these other 2 new regions, as well as this one called SCAN7 is really way up. FastX is really down. That's done using RT-PCR. And here's their northerns showing the same thing. What they're doing is they grow their cells in CDM, 3 different ODs, and THYE, 3 different ODs, showing the expression, and that should match what they see from the previous slide. So this confirms that these things are expressed. This is why I'm saying this is I think this is better where they actually do this experiment, because this is confirmation of your small RNAs. And that's that paper. The last one is this cowbug. Aglactiae. So they did this experiment on 3 different strains. A human strain and 2 cow strains. Now what they've done was come up with a number of reads. So this is the Illumina data for all of their reads. And they've done it in milk, broth, all kinds of conditions. They'll want to compare the milk with the broth for the bovine strain, and milk with the broth from the other bovine strain, and milk with broth from the human strain.
Okay.
Next, what they're doing is they're showing the upregulated genes. So there's lots of upregulated genes when you're comparing these two. This is the first figure, so I think there's about 5 figures here.
Okay.
What they're doing is they're showing you growth curves, and they're doing the growth curve for the NEAM 316 in milk and THYE and bovine in the same conditions, and the human one. As you can see, the NEM 316 does much better in THYE than it looks like. Actually, milk does much better in milk than it does in THYE. The other 5 growth curves all come up to about the same CFU per mil from right to reader. And in the next picture, they're showing all the different classes of upregulated and downregulated genes. Very similar to the last paper, but they're clustering them based on their biological processes. Same thing on the next slide. They're clustering them with biological processes. Okay, this is showing a difference between strain 1 and strain 2 grown in milk and broth. They're also looking at the lac genes because it's a virulent pathogen, so they want to study the difference, and they show what they want to see if there's a difference between milk and broth. And look at this for the LAC-R for NEM 316, there is. And actually for all of the other genes, it's different between milk and broth for NEM 316. So in broth, you don't get the upregulation of those genes, but in milk you do.
Yeah, because
except
they need
for
to
LAC-R.
metabolize lactose, right?
Yeah.
Which makes sense.
Yeah, it does make sense. It makes good sense. In broth, you don't really need it, but in milk, you do, and that's why you get these white and blue bars up on top.
Okay. Look, this is just a big figure.
And
the only figure, actually.
only real data, yeah.
Yeah.
And here's a figure where what they're doing is they're showing the phylogeny of these genes. So they're including a bunch of different streptococcal genes or organisms. As you can see, here's mutans.
Oh, I see.
So it's on the list. So they've clustered 4 different groups based on their phylogeny. I know this slide says class 5, but it's really class 10.
Oh, what's that? Sorry?
I got the wrong thing there. That's that paper. Any other questions?
No, great job, Andrew.
Yeah, okay, thanks.
Mastitis. Oh, okay, that's why they looked in the